Ever wonder how to analyze mountains of RNA-seq gene expression data to find meaningful insights without having coding experience? Today we're diving into GEO2R, a powerful web-based tool that makes these complex tasks accessible to everyone. So why reanalyzing public RNA-seq datasets? First, there is no need to generate the data, which is usually time-consuming, expensive, and difficult. For example, if you have to find human samples. Second, when you need to validate your experimental results, you need to generate or narrow hypotheses before designing experiments or when applying for funding. In this guide, we will explore what GEO2R is, what kind of data it handles, how to use its features, interpret its results, and understand its capabilities and limitations. GEO2R is an interactive web tool designed to help you compare two or more groups of samples within a GEO series. Its primary goal is to pinpoint genes that show significant differences in expression across various experimental conditions. The results are presented in a clear, organized manner, typically a table of genes ranked by their p-value alongside various graphical plots for visualization. At its core, GEO2R leverages robust R packages from the open-source Bioconductor project. GEO2R is versatile, handling both RNA-seq and microRNA data. For RNA-seq data, it utilizes the DESeq2 package. This is crucial for identifying differential expressed genes using NCBI computer raw count matrices. DESeq2 is known for its consistent performance even with varying study sizes. Let me show you how GEO2R works. Let's find first GEO datasets. As we mentioned before, GEO datasets contain next-generation sequencing data, including RNA-seq, ChIP-seq, and microarrays, among others. For this example, I'm going to use an Alzheimer's disease dataset. So we can narrow the results by study type. In this case, we are interested in expression profiling by high-throughput sequencing, which is how they define RNA-seq data. Then we click on Show, and we click on the link to filter by this kind of data. Second, let's find human data. So under Organism, click on Customize, and select Homo sapiens and Show. Then click on the link to activate the filter. So notice that you can filter from higher to low number of samples. And now we have studies with 764 samples, 625, etc. We're going to use this dataset to show how GEO2R works. So make sure first that this is the dataset that you are interested, that the samples were collected properly, evaluate your inclusion and exclusion criteria, and when those are met, select Analyze with GEO2R. So the first thing to do is to define the groups that you want to compare. For this example, we want to compare female with Alzheimer's disease versus female controls. So first, I'm going to select the Alzheimer's disease samples, female. Notice that we can organize by sex. So we select those by simply clicking and dragging, and then clicking in the group. And then select the control group. Click on the sample and click on the group that the sample belongs. Okay, so we have 20 females with Alzheimer's disease and 20 female in the control group. Now we scroll down and notice that we can analyze samples, or we can go to options and define our thresholds, our cutoff thresholds. In this case, we're going to get every differential expressed gene that is significant for the FTR 0.05. And then click on Reanalyze. So here are the results. You get the volcano plot, which you can explore and download. See, notice when you mouse over those dots, you can see which are those. For example, we have one here. In this case, a pseudogene. So you can see which are those differential expressed genes, upregulated or downregulated one. So there are other plots, including the PCA, and this diagram that shows you can explore and download. In this case, Alzheimer's disease compared to control. You can download the significant genes. Containing the fold change and the minus log 10 of the adjusted p-value. You can also download the full table with all the differential expressed genes. So here is a table containing all differential expressed genes. Once you have your differential expressed gene, you can continue with the downstream analysis, including pathway analysis.